So, as was indicated, we have the early translational grant for Parkinson's disease. And rather than kind of indicate what the incidence of Parkinson's disease is in the community, I think it's fair to say that everybody knows somebody with Parkinson's disease. And if you don't know somebody personally, you know some major figure who has Parkinson's disease. It's characterized by freezing movements called bradykinesia and then tremors. The pathology emanates from the suspension nigra where dopaminergic neurons are made, and these have to project to the striatum, which by the nigrostriatal pathway, and this controls movements. Now, the early translational grant was predicated on some work that Gene Redman and I published back in 2007, in which we were able to show that human neural stem cells, in what amounts to the most authentic animal model of actual human disease, in other words, an MPTP intoxicated monkey, was treated initially with undifferentiated human neural stem cells. I won't go into the details of the model except to say that there is no model, animal model, that is as close to the actual human disease in terms of not only replicating the pathophysiology, but also the side effects, and the behavior can be scored using actual human scales. So a very informative model, and in fact, it was this model that was the basis for fetal tissue transplantation back in the early 1990s, also for the trials documenting the efficacy of deep brain stimulation. So what we initially did, and this is emulated again in our early translational grant, was to put the cells both in the nigra and the striatum, and what we found basically was, based on the Parkinson score, so a high score means a lot of Parkinsonian symptoms, that in the animals that were transplanted, they weren't normal, but there was a dramatic decrement in the degree of Parkinsonian symptoms. And we found that this functional recovery was based on at least 3 different mechanisms. One was actually some degree of dopaminergic neuron replacement, as indicated over here, but a small amount. I'd say only 3 to 5%. So clearly not carrying the bulk of the therapy. A huge amount was carried by preservation of the animal's own dopaminergic neurons in the nigra and their projections to the striatum. So preservation of the intoxicated dopaminergic neurons, preservation of this pathway. And then finally, for mechanisms we're still trying to figure out, these Lewy bodies, that's what alpha-synuclein aggregates are commonly called, seem to be diminished in animals that receive this. With that in hand, we recognize that there are many ways since 2007 to get to the neural stem cell. And we had to figure out, if neural stem cells are useful, what is the best way and can we do better? Is it through the fetal route? Is it getting from the adult CNS, through ES cells, through iPS cells? And we knew that we could start back over here and get to early neural stem cells, later neural stem cells, and by bootstrapping our way through mechanisms that emulate development, we could get to dopaminergic neurons. So the purpose of the early translational grant was to determine and to actually do the very first head-to-head comparison ever done on all the candidate stem cells that, at least at the time of the grant, were out there in the literature as being plausible candidates for use in any neurologic disease. So these numbers simply represent the head-to-head comparisons that we would be doing to get to human neural stem cells, and then from human neural stem cells, either leave them that way or get to ventral mesencephalic precursors. So these are the 6 candidates, and they're the logical ones, starting with ES cells and dopaminergizing them, starting with iPS cells, dopaminergizing them, going to the fetal ventral mesencephalon and implanting that. And, of course, the key is having excellent control cells. And these are a list of the control cells. Notice that human mesenchymal stem cells were a negative control, at least in this particular disease. Aim 1, and I won't go through the details, would be to take all of these and characterize them in vitro through all the reasonable characterization steps. Their karyotype, their growth potential, what do they become, can they make dopamine and dopamine precursors, what is their transcriptomic and proteomic profile. And then the most important thing was, of course, how do they do in an actual animal model of Parkinson's disease after long-term observation, at least 12 months, certainly no shorter than 6 to 9 months. And one assessment would be how well is function of the nigrostriatal system preserved. And there are a lot of ways to do that. Interestingly, behavior, a Parkinsonian score, and I'll show this later on, correlates excellently with actual biochemically determined dopamine levels. The next is, what do the cells do? Where do they become? How long do they survive? And do they become dopaminergic neurons? Do they become tumors? Do they become inappropriate cell types in the brain? And then finally, looking at the interaction between the donor cells and the host cells. So the progress that we've made, and we're starting to get to the end of our 3-year early translational grant, is that we've transplanted close to 100 monkeys, accumulated over 10,000 observations, and most of these animals, some of them we did short-term observations just to see what the cells were doing. Most have had long-term observations up to 20 months after injection, and that's important to actually see not only for safety, but that is a long period of time to restore function to the nigrostriatal system. We demonstrated that across observers there was no inter-observer variability, and as I mentioned before, this now constitutes the largest and most comprehensive head-to-head comparison of stem cells in any model of any disease, let alone a large animal model of Parkinson's disease. And after all is said and done, we've come up with a developmental candidate. And what I wanted to do is sort of say very quickly, how did we come to the candidate that I'll indicate to you? And let me see, I think I even gave it away. Nope, didn't. So one was to compare all of these stem cell candidates head-to-head, looking at the Parkinson score and the healthy behavior score. Now, interestingly, we first documented that this behavioral score actually correlates very well, and this has been known in the literature, with actual dopamine content. So that allowed us to actually accelerate our assessments. Once we had the candidate, of course, what we would do is then advance them to IND-enabling studies. So the first the candidate was simply undifferentiated fetal CNS-derived human neural stem cells, sometimes even trying to augment the environment with GDNF. Most of the cells, as we demonstrated in 2007, became neurons, but not dopaminergic neurons. Not that that was inconsequential, because they would follow the host nigrostriatal pathway and actually serve a homeostatic and preservation effect. If we tried to dopaminerge those cells, and one can do that with an astrocyte-conditioned medium, in vitro and in vivo, one got more Th-positive dopaminergic cells. However, if you looked at the behavior, and let me tell you, this is what one does. This is the Parkinson score, and one will look at the slope. Where do you start on the Parkinson score, and does it fall? Do you have a really deep slope? In other words, the Parkinson symptoms have diminished, and you can see that not a very deep slope, and in fact, one of the animals actually seemed to not get better, or at least get worse. So while neuroprotection might be important, we thought that we could make better impact if we were to increase the production of A9 dopaminergic neurons. So one way to do that is just to go to the fetal ventral mesencephalon alone. Just go out, and they're already on their way to becoming dopaminergic neurons. That seemed plausible, and some of the animals did get better, but a large number of the animals did not get better. Some of them actually did not have sustained recovery. So then we started going to developmental biology, which also is following this field as the stem cell field was growing, and recognizing that FGF8 and sonic hedgehog is a ventralizing signal, FGF8 is a mesencephalic instructing signal, that maybe that would help give us more A9 dopaminergic neurons. So we went to iPS cells, dopaminergize them, but subsequently we started learning, the field started learning, that this is not a complete dopaminergizing protocol. And in fact, we got some improvement, but not a whole lot of improvement. So this seemed to be, and in fact, in some of the animals, actually a positive slope, so the animal's getting worse. So could one try a better, and then this simply is using that exact same protocol on ES cells, sonic hedgehog and FGF8. one could get nice Th-positive cells when one engrafted, and about 25% were Th-positive. But still, even though we were getting desirable slopes, not as good as it could be. The improvement was moderate and variable, and here was a hooker. We got in one of the animals, prior to dopaminergizing, we actually in one of the animals got an accumulation of something that looked like a tumor. So clearly a red flag, and made, at least using this protocol, a no-go for us because of the tumor risk and the less robust behavioral outcome. With the caveat, however, that because some of the animals did get better in an encouraging way, if we could have a more complete dopaminergizing regime, and if we could have unambiguous safety, then we would actually have a pretty good product. And that involves some more developmental biology, recognizing now, and this is thanks to some work done by Lorenz Studer, that if you actually instruct the cells to become floor plate, that is where the dopaminergic neuron precursors come from in the ventral mesencephalon. So now creating floor plate through Wnt inhibition via GSK3 beta inhibition, now we got lots of dopaminergic precursors that engrafted nicely, gave us nice dopaminergic neurons implanted, and really nice, very appealing kinds of curves. So very deep improvement in Parkinson's score, much better than in the other candidates. This shows the degree of change compared to the other candidates head-to-head. This illustrates that compared to controls, an improvement in healthy behaviors. So now we had our candidate, but there was still this risk that we had to contend with. How could we get rid of the risk? And the risk would involve absolutely making sure that the cells stayed in a ventral mesencephalic lineage. That would be a positive selection, and then excluding anything that did not fall there. And again, this came down to developmental biology, putting in some safety selection markers in the differentiation pathway. LMX1A early on, NER1 early on. The cells in their developmental pathway do express these. So it's possible to do that. We now knocked in reporter genes, or our collaborators in Australia knocked in reporter genes by homologous recombination in a very benign way. This assured that the cells were absolute, that the cells we pulled out were only in this lineage, and actively excluded anything not in that lineage, including even primitive neuroectodermal cells, certainly teratomas, anything inappropriate there. This then would ensure reproducibility of the cell population, safety, purity, and a high degree of dopaminergic neurons. This simply indicates that they really were knocked in just to exon 1 in those sites in a very benign way. This indicates that if you fact sort these guys to select, you still get cells that are exactly what you want in the A9 dopaminergic lineage that will continue to give very nice neurons, which means now this is our candidate, and this is what we would like to advance. Human ventral mesencephalic precursors generated from ES cells, the H9 line, using a protocol that confirms floor plate identity and with a selection, both for safety and for locking lineage commitment based on ventral mesencephalic gene expression. This is clearly compatible with expanding cell, certainly getting through regulatory hurdles. This candidate clearly has very convincing, statistically significant, reproducible, consistent, sustained improvement in a large animal model that absolutely emulates the human disease. Having these selection markers seems entirely benign. So far, it hasn't altered their behavior either in terms of safety or in terms of efficacy. There's been no immunogenicity, at least as we prepare the animals. And we think we know exactly how we want to administer the cells, when we want to administer the cells, and the mechanism of action seems to be dopamine production and synaptic release into the striatum. And in fact, dopamine release and the production of dopamine and its metabolites would be part of the release criteria, as well as the normal logical steps that one would look for in terms of assuring purity. We think we know how to scale these up. We think we know how to administer them intraoperatively, particularly using a catheter that is flexible and multi-pronged that it was developed by Daniel Lim with some funding as well, and with whom we're collaborating. So I think I'm running out of time, but our next step, and this brings us almost towards the end of our early translational grant, is now to launch filing an IND and getting ready for a clinical trial. And this is about what we're getting ready to do now. These are, I'll just put these up, but these are everything that everybody would recognize is what are the pre-IND and the IND enabling studies. We're on track to have a pre-IND meeting with the FDA. And we're fairly confident that we can now take this product and in maybe 2 years get through the regulatory hurdles and launch at least a phase 1 clinical trial in a disease that, as you know, is absolutely incapacitating, particularly because even the well-known therapeutic agents ultimately stop working, and hopefully we'll be there when that happens. So thank you very much.